Hi everyone, I hope all is well. My name is Saad Razaagarzadeh and I will be presenting our research on an augmented reality-based tumor localization and visualization method for robot-assisted breast surgery at the 4th IEEE International Conference on Human and Machine Systems, Toronto, Canada. So the outline for my presentation is as follows. I'll be going over a bit of background and motivation of this research, then going into the methodology itself, followed by experiments and results, and finally the conclusions and future work. So breast cancer is a major concern. A lot of people are dealing with this. It's the #1 cancer in women in Canada. 1 in 8 Canadian women will be diagnosed with cancer in their lifetime, and 1 in 36 will die from it. You can see some more details of these stats on the right, and this is not limited to Canada alone and has caused close to 700,000 deaths in 2022 alone. So what are the treatments for breast cancer? There are chemotherapy, radiation therapy, hormonal therapy, immunotherapy, and surgery. And when going over surgery, we tend to give 2 options, and the surgeons either perform mastectomy, which is the complete removal of the breast, and you can see in the image the outline that needs to be removed. And secondly, then the option is lumpectomy, which is the removal of the tumor and the surrounding tissue. So instead of removing the complete breast, the surgeon removes only the tumor itself and the tissues around it. So lumpectomy is often preferred because of cosmetic reasons, but the challenge is to accurately locate the tumor, because if the surgeon wants to cut the breast and in the surgery take the tumor out, there needs to be a precise localization of where the tumor is. And this is leading to the current localization methods, which is either wire localization, where a needle is placed into the breast preoperatively, so before the operation itself. And you can see in the image here that the breast is compressed and the needle is placed so that the end point of the needle is sitting at the position of the tumor. And this may need to be done a day before the surgery, and the patient will need to have this needle sticking out of the body, which is definitely not a convenient option. And there's non-wire localization methods as well, such as radioactive seeds and magnetic seeds, but these are invasive as well and needs to be placed into the body before the surgery. So because these methods are invasive and have patient discomfort, the question that we are trying to address is that can we develop a method to localize the tumor, which is non-invasive before doing the operation, it's high in accuracy, and it allows for a 3D visualization of the tumor for the surgeon. To address this question, we'll go into the methodology of what we are proposing. So before going into what we have done, I'll spend a couple slides on a bit of background on robot-assisted surgery itself, and also augmented reality, which are the 2 main elements we use in this research. So in robot-assisted surgery, instead of the surgeon doing the operation by hand, there have been robots that have been developed, such as the Vinci surgical robot. So you may have seen this famous video of the surgery being performed on a piece of grape, where it involves both cutting inside and also peeling the surface membrane of the grape as part of the surgical operation. So this Da Vinci surgical system has been used for more than 10 million surgical procedures, and more than 60,000 surgeons have been trained to use this. And the question that is arising here is that why do we want robotic surgery? So the 3 main advantages of robotic surgery is its enhanced precision, the intuitive maneuverability of the robotic arms, especially in minimally invasive surgery, because these robotic arms are going to enter the human body, which take a lot of less space than open surgery, where instruments are inserted using the human hand. And finally, the improved vision that is as a result of the stereo vision feedback is another advantage of robot-assisted surgery. So now going into the other elements that I want to talk about is augmented reality. So AR is a technology that projects computer-generated augmentations on top of reality, which allows for a better efficiency and situational awareness. And it's not only limited to surgical applications, but AR is a technology that is used in a lot of domains. So some applications of its use in surgery is orthopedic surgery, neurosurgery, and minimally invasive surgery. And the main benefits of it is allowing for an enhanced spatial awareness and minimized invasiveness with increased precision that comes as a result of it. So to address the initial question that I proposed in the motivation, and now using the fundamentals that we have seen, in our framework, we want to initialize the position of the tumor using medical imaging in the breast, while the breast is in a resting position in the operating room, just as the patient is laying down for the operation. We then superimpose the 3D model of the tumor on the physical breast and display it on the Da Vinci surgical console, and the surgeon will basically be able to see this augmented tumor on top of the physical breast, which allows for seeing essentially where the tumor is without needing to cut open the breast. And finally, and more importantly, because the breast is highly deformable, as a response to different forces and different movements of the breast, and while the patient is laying down, there will be deformations to the breast which lead to the movement of the tumor. So we also propose a distance-based interpolation algorithm to update the position of the tumor in real time to these deformations. So the architecture is seen as follows. There is an initial registration phase where the initial position of the tumor is detected using ultrasound data. There's a surgical scene, which is the phantom itself. I have the experimental image on the right-hand side here, where the markers placed on the phantom would be seen by this ECM cameras, the endoscopy camera manipulator arm of the Da Vinci robot. And the two ground truth cameras that you see will detect the actual position of the tumor. And these all go into the Unity app of allowing for the tumor interpolation algorithm to update the position of the tumor and overlay in real time. Now, as part of this methodology is the camera calibration process, because there are two cameras as the ground truth cameras and two endoscopic cameras as well, which need a lot of calibration to find the relationship between the transformations of these cameras, and then building the Unity app itself, which allows us to have the two displays of the ECM camera on the surgical console. And this allows for a 3D vision because we have two different cameras at two different locations. So next we have the interpolation algorithm itself, which would basically have the position of the tumor equal the position of the tumor at the initial time, plus the deformations that come as a result of the deformation of the markers on the surface of the tissue. So if I play this video here, this animation gives a good idea of how the movement of the surface, which results in the movement of the markers, would lead to the movement of the tumors. And this red tumor would be the updated tumor, and the green tumor is just the initial position of the tumor. And our proposed interpolation algorithm would give a higher weight to the markers which are closer to the tumor, and less weight to the markers which are away from the tumor. And this would allow for a distributed weight in the effect that they have in the movement of the tumor as a result of the deformation of the surface. So going into the experiments now, I've shown the experiment setup before, but you see the image of the phantom as well. We made this using gelatin, and on the left-hand side is what the surgeon sees on the console. You can see the green blob here is the actual tumor, and this red superimposed blob is the augmented tumor. So for the first experiment, we placed the phantom in the surgical workspace, and the goal was to just detect the accuracy of the marker detection and tumor overlay using the ECMs for different initial tumor positions. And then we found a root mean squared error for the X, Y, Z directions, and found the RMSE of 6.37 millimeters for these three different initial positions. And this image in the middle shows where the ground truth tumor was, the green, and where the AR superimposed tumor landed, having this medium or the mean of 6.37 millimeter difference. For the second experiment, to validate the interpolation algorithm, we had three different deformation scenes where force was applied to the tumor from three different locations. In figure A here, we have force being applied from a position near the tumor. In figure B, we have a mid-range of a distance where the force is applied, and finally force being applied from further away in figure C. And what you see here is the displacement of the tumor as a result of the force as seen in the ground truth cameras, and also where the augmented tumor has moved. So 4.4 millimeters, 4.8 millimeters, and 4.2 millimeters is the approximate RMSE that we see for these three conditions. Now, to conclude this presentation, I would like to summarize that we propose an AR-based tumor localization and visualization for robot-assisted breast surgery, and we also propose an interpolation algorithm to update the position of the tumor in real time as a response to the different deformations. And finally, the future work in this area would be exploring the markerless approaches and also finite element-based methods for estimation of tumor movements, and also exploring the effect of breast tissue cutting to see how cutting the tissue would affect the overlay, deformation, and the movement of the tumor itself. And lastly, the use of other means of display of AR, such as head-mounted devices, such as the Microsoft HoloLens 2, for visualization of the tumor and the deformations. Thank you very much for your attention. I would like to also acknowledge our teammates at the Telerobotic and Biorobotic Systems Lab, and also my supervisor, Dr. Mahdi Tabakkoli. Thanks again, and I'll be happy to take any questions.